Thank you, Michael. Well, I also want to thank the Institute of Medicine, the organizing committee, and all of you for coming. And I think Michael has set the stage beautifully, and I hope we have a day and a half of important discussions, because we do have to sort out what is the proper use and how to optimally take advantage of naturally occurring cancer in pets to maximal benefit both to human patients and obviously to pets as well. So my first slide just reiterates the problem, which is, and Michael touched upon this, the cancer drug attrition rates are actually significantly higher than any other therapeutic area. And just to give you an idea, most recent statistical data, about a 5% success rate of drugs being developed for anti-neoplastic compared to 20% success rate in the cardiovascular drug development field. So I'll show you in a few slides. That's a lot of money, and that's a lot of failures. And I don't think we know what the right failure rate is, but I would submit that 95% is a bit too high. And obviously, this has led to many attempts to improve outcomes. And I'm not going to get into, there's been a lot of dialogue recently about improving statistical methods. So Bayesian methods versus standard methods, and you know, better use of models, better use of iterative, you know, from the clinic to the lab, back to the clinic, better zebrafish models, better mouse models, PDXs. Suffice it to say, my own personal opinion is it's not a problem of statistics, and I'll show you some data to suggest that. I think statistics could potentially help us to expose less patients to ineffective doses, but that's not even clear because recently there's been some modeling that suggests if you do a Bayesian model, you could actually end up exposing more patients to the ineffective arm. There's some posterior probability assumptions that aren't always right. So the real question is, how do we get this number to a more acceptable success rate? So this is already 10 years old or so, but it hasn't changed a lot. And the problem is that most drugs fail in the late stages of development, even more so in oncology. And so this just shows, and I have a few slides that will illustrate this in various ways. But 70% of oncology drugs that enter phase 2 fail to go to phase 3. Now, that's important because there's a big step up in resources going to phase 3 in a much larger number of patients. But I would argue that that is higher, and I would say that most of us felt as we got into the era of targeted drugs that would be lower. And I have, again, we don't have the most recent data, but it's not changing as rapidly as you would guess. The other interesting thing is that once you get to phase 3, where you're making a huge investment, you know, 60% of those drugs then fail when they get into phase 3. And so late stage failure, and I'll go over the stages for those of you who are not totally conversant with phase 0, phase 1, phase 2, phase 3. But that's an enormous risk and an enormous cost. And the other thing that was interesting is to find out that it's usually not toxicity, it's usually the drugs fail because of lack of efficacy. And so that's really a very substantial problem. So given our state of knowledge in 2016, our understanding, or 2015, our understanding of the background, the genetics, targeted therapy, what's the problem? And so this is from a paper, again, about 10 years old, from Nature Reviews Drug Discovery. And here I show this slide because this compares oncology compounds to all other drugs. And you can see that it's not dissimilar about 61 to 63% of drugs that go into phase 1 reach to phase 2. But look at the drop-off. 28% of oncology drugs that get into phase 2 make it to phase 3. That's about, almost 40, that's about a 40% drop-off compared to non-oncology drugs, where it's about a 40% going from phase 1 to phase 2. And then it continues to drop off. There's another drop-off of less than half of drugs going into phase 2 get into phase 3, whereas in other non-oncology drug development, it's almost 60%. So there's another 20% drop-off. And then once it gets through phase 3, it's really still 30% of those drugs aren't going to get to registration. So this is an enormous cost of dollars, of patience, and of people time. And so, again, I don't know what the sweet spot is, but I think why oncology is so much less efficient than other areas of drug development, I think is an issue that the field really has to grapple with. And to put it in another perspective, here's the number of drugs in development as of 2010. And the big C, you can see more than twice as many drugs are in development in cancer than in virtually any other system. So you could ask, why is that if the risk is so high? But obviously, pharma has figured out the model and they can still make money. But this is at an enormous cost to pharma, to our healthcare system, to our patients, to all of us. And so, this is an enormous pipeline with an enormous failure rate. So this has really been termed the valley of death. So what happens? Why can't we get, in a much more efficient manner, drugs that get past, and I'll show you in a minute, there are thousands of preclinical screenings that get a drug to go into a patient to begin with. Why do we fall? And why is there this enormous valley of death? And so this slide just shows the just getting a drug to clinical trials. You can see that it's been estimated that, screening 5 to 10,000 compounds, you maybe get 250 that go into preclinical development. And of those 250, 5 get into phase 1. And so we just can't afford to get from 5 to 1. It's just not, it's just not acceptable anymore, I think. And the other thing I wanted to show, if you look at the number of patient volunteers, because after all of our patients that go into phase 1, phase 2, and phase 3 studies are volunteers. They are patient volunteers. They're an enormous resource. And you can see the ramp up, 20 to 100 patients in phase 1, 100 to 500 patients in phase 2. And when you get to phase 3, you're talking about in the thousands. So you're talking about basically a log ramp up. And Michael already mentioned the timeline. It's typically more than a decade from drug discovery or preclinical discovery to getting a phase 3 study. And then it's once the phase 3 gets done, you can see it's usually once you get into registration, it's relatively quick. So this is now just showing the cost, and it's been estimated that overall the cost, I think Michael mentioned this, it's in the hundreds of millions of dollars. But this, just looking at preclinical screening, there's already an investment of $1.5 million to come up with a candidate. So from there, the costs just really ramp up. And so my point, or at least the question we need to ask ourselves is, once we get into a clinical candidate, how can we not lose so many drugs? How can we ensure that the drugs that get into clinic, have a better success rate? I don't have the answers. I'm just posing the question, and I hope this discussion over the ensuing day and a half will give us some ideas, because I think the theme of this meeting is we have not utilized companion animal or pet animal studies maximally, and I'll pose some questions why that may be. So this was from a recent review by Paul Workman's group, with the sort of the optimum, or I would say the optimist view that things are going to change now with targeted therapy, that we have a much better way of doing, picking a target, a kinase. Those are the easiest targets for now. So we pick a target, we do hit identification, we're very good at that. Then you do medicinal chemistry, you get to lead identification, you do lead optimization, you then do three-dimensional, and you can talk about in vitro three-dimensional, and we certainly are doing more PDXs, what's the right model in mice. But for those of us who have been around a long time, xenografts didn't solve our problem, unfortunately. So we do the studies, either three-dimension or mouse xenografts. We then further narrow down. We eventually get a drug into a patient. Sometimes we'll take a phase 1 and go back to the clinic, revalidate. So the idea or the hope is that this is a more efficient model, because you're crisscrossing back and forth between preclinical testing, preclinical models, and refining your molecule, refining your approach, refining your targets. But the fact is, we're still failing miserably. And so one of the issues that I would, in my opinion, is that where we've really failed the most is in developing biomarkers, selecting biomarkers that will help us select patients that are either not likely to respond or more likely to respond. So negative biomarkers to screen out patients. And a perfect example of that is the development of Herceptin. And so for those of you who don't know the whole story, this was the antibody developed against HER2. And it turns out that a number of patients with breast cancer have amplification of HER2. But the first study done with Herceptin in breast cancer patients, the response rate was pretty bad, 20%, 15%. There was no selection. Once we identified that it's the patients with amplification of HER2, now that would respond to blocking that, now the response rate went way up. But that is one of the precious few examples of where we actually got a positive selection biomarker that allowed us to enrich. So enrichment strategies are becoming extremely important both to academia and to pharma. And I would basically say that most drug development plans, absent a biomarker, people are very hesitant to move forward. And I think that's a good thing. But the problem there has been our ability to identify and accurately select biomarkers moving forward are relatively few. I mean, we have a few. I think the companion diagnostic of BRAF mutation with the development of vemurafenib is a recent case in point. But you can count on one hand the co-development of a really good biomarker to select and match the right patient. I mean, this is a field that's exploding, but we are lagging way behind. And one hope I would have is that companion animal studies may help us in biomarker development, and it really hasn't been used very well at all. So this is a slide I just borrowed from the Division of Cancer Treatment and Diagnosis. This is the NCI Experimental Therapeutics Chemical Biology Consortium pipeline, and this is the idea that the NCI has to try to go from identifying a target, trying to screen for is it a valid target, using high throughput screening to optimize a particular compound, using medicinal chemistry, and then developing a lead compound and getting it into clinical trial. But really what I want to focus and emphasize here is the use of both transgenic and patient-derived xenograft models. We've not optimally figured out how to use those, and each of those mouse models, I would suggest, can ask different questions. One of the things that we also seem to do particularly bad at, and I can tell you I have unfortunately too many personal experiences with this in my own practice, is pharmacodynamic endpoint validation. So sometimes we get a drug that we don't have really good PK, and it looks fantastic in a model, but the PKs fall apart in the patient. And we don't really have, so what a pharmacodynamic endpoint is looking at a target that matches the PK, so it's even a step up. And we don't have a lot of really good examples of that. Obviously, the use of small animal imaging is something that we're all hoping will also help us with pharmacodynamic endpoints. But as I mentioned, the biomarker assays are really going to be key. And one of the other issues that I don't think academics fully appreciate is that a terrific biomarker in your laboratory with xenografts or animal models in no way is going to be a robust clinical biomarker. It takes a huge ramp up and step up to go from a Western blot that may help you decide the cell lines or the xenografts that are going to respond, to being able to use this in a clinical setting. And really, it's biomarkers that I feel the field has fallen woefully short in. And of course, we all hope that as we move into next generation sequencing, and there's been an enormous effort between industry and the FDA and academia to figure out how do we use genetic markers, something again that Michael mentioned. And the FDA is being extremely flexible to figure out how we can use this ever-increasing sophistication to genetically characterize something, again, to help with biomarkers, but it's tough, and we have a ways to go. So you may see this slide again from Chan, I'm not sure. But this is sort of the conventional development path. So as I mentioned and Michael mentioned, we have these preclinical models. We then usually do toxicology in beagle dogs or non-human primates. We do some small animal modeling, and then we go into phase 1 human clinical trials. So phase 1 human clinical trials are in the classic model designed to identify the dose to move forward. So it's to identify the maximum tolerated dose. So I want to just digress for a minute and state that I started my oncology career in 1983. And I will tell you that in 1983, the expected response rate of phase 1 clinical trials was 3% or lower. And I would say, and all we were looking for was to get probably not as much pharmacokinetics as we should have gotten, but just to define the maximally tolerated dose. And it was totally empiric. We put 3 patients on a dose, and if if none or one had, if one had a toxicity, we expanded. If none, we went to the next higher dose, and we kept escalating until we got toxicity. Now that paradigm doesn't work anymore. And in fact, I would submit to you that the clear distinction between phase 1 and phase 2 is gradually disappearing as we develop drugs now targeted against a particular mutation where at least we're looking for patients, or we're enriching for patients with a mutation, and we're looking to see, is the drug affecting that? The first wonderful example of that was imatinib in chronic myelogenous leukemia. And I remember when Brian Drucker reported his first report was actually a poster at ASCO, where he had about 80% response rate in a phase 1 study. And I can tell you that was unheard of. But in retrospect, as we've gone back and looked over the last decade, we are expecting to see responses in phase 1 studies now for most of our. So that's a step forward. and yet we're still not getting from the early responses in phase 1, we still have this enormous attrition rate. And so traditionally, phase 2 was where you had the maximum tolerated dose that you then brought into target tumor types. And again, this has dramatically changed because it used to be the target tumor types were colon cancer, lung cancer, breast cancer, and you would put, often we did what was called a Simon two-stage design where we would put 9 or 11 patients on, and if you saw no responses, you would stop and say there's less than a 20% chance that this is going to have a 30% response rate, or depending on how you did your statistics. But basically, you were looking for a signal in a particular tumor type. As I said, now with more targeted therapies, number one, we're looking for responses earlier in phase 1. We often do what's called phase 1/2. We're also not necessarily doing tumor types, although there's a lot of discussion. But often if you have a drug targeting BRAF, for example, maybe you should be treating, or even better, Herceptin, which targets HER2. We now know that HER2 can be overexpressed and amplified in gastric tumors. So rather than just treating breast cancer, you might want to treat across histologies, at least if you have an enrichment strategy, and then ask, do different histologies respond differently or the same when they have the same target that the drug is going after. And the other thing that's changed is the maximum tolerated dose. Many of our biologics, some of our antibodies, we never reach a maximally tolerated dose. We reach an effective dose, and we just stop escalating. So the whole paradigm is changing. And then obviously when we get to phase 3, which is again where we have an enormous fall off even to the current time, now we're looking for improved outcomes. So we're generally comparing this drug to standard of care in a randomized fashion. And obviously the best marker is overall survival, but we can argue, and there have been many other endpoints that people have gotten drugs registered for recently. So what is the reason for the high attrition? First of all, I would say that we know now that cancer is an extremely complex problem. The preclinical models, as I've said, and I hope I've convinced you of, are clearly not as predictive. The pathway so far, our standard pathway, is linear and ignores more of this iterative back and forth, and we're starting to see that, but it really hasn't come to full practice. And so you've seen some of these pictures already, and I'm sure you'll see them again. So what is the promise of comparative oncology? Well, you know, patient-derived xenografts are interesting because they take the complexity of the human cancer, but they put it in a microenvironment that's foreign. They put it in a mouse microenvironment. Genetically engineered mouse models are terrific for answering a specific targeted problem where every tumor cell is driven by a particular genetically developed tumor that we engineer. But often we don't get the complexity that we see in naturally occurring cancer. And so to me, one of the promises of comparative oncology is that these are naturally occurring evolving cancers that can study the complexity and biology of a naturally occurring tumor. Michael already went over this. We have an enormous volume of pets to draw from. And again, just to remind you, we're talking about 1 million pet dogs diagnosed with cancer a year. And obviously, you know, most of us have owned pets during our lifetime, and we're pretty heartbroken when our pet starts dying of cancer. So if we can help pets and at the same time improve our pipeline, I think it is a win-win, as Michael suggested. And I just highlighted in red here, you know, the specific obvious advantages are the tumor heterogeneity, spontaneous metastasis, and recurrences and resistance. So this is a model, again, this I think is from a review that Chan and colleagues wrote, but where could we move pet animals into this sort of linear, and here are some ideas. So we stood up the comparative oncology program about 10 years ago, and I was fortunate to have met Greg McEwen and Steve Withrow and Chan all within about 6 months, and it really had an enormous impact. These were dedicated professionals interested in cancer, and we just felt, we need to integrate the fields much better. And so one example would be if we have 3 or 4 analogs, and this has happened and been tested in the comparative oncology program, and it may be discussed. How do you know which analog to move forward? They all have slightly different properties, but they're pretty similar. They're all targeting the same thing. We pick one, but maybe we don't pick the optimal one. And here's where going back to a dog model might help us pick a more potent analog to move forward. Certainly in terms of phase 2 clinical trials, again, there's an opportunity to be more specific about regimens and doses. And I can't tell you how many drugs have gotten all the way through phase 3, and we don't really know the best schedule. And it takes us years to sort that out. And here's an opportunity to really focus a little bit more on schedule and biomarkers. And even in dogs, as you heard, there are multiple histologies, and it might be a chance to compare several histologies with the same genetic markers and tell us, do the histologies, at least in dogs, behave similarly or different. And I can tell you with BRAF mutations in colon cancer, they behave totally differently than BRAF mutations in melanoma. We now know why, but there's a mechanism. And if you try to treat BRAF mutant colon cancer with vemurafenib, you'll see no responses because the wiring's different. And so I hope that these are some of the areas that we can think about that could actually get us so that we have less attrition as we move through this, especially as we get into phase 3 studies. So, I think it's our hope that maybe we can bridge this valley of death, and maybe companion animals can help. Obviously, it's not going to be the sole answer, but I think there's a real untapped opportunity here. So I'll stop here. I don't know if we take questions now, Michael, or just wait till the panel discussion.
Cheryl, should we take questions now or just do it during the panel?
Okay, that's fine.
Just save it for the panel. But we have a little extra time in this session,
Yeah.
so I would say if there's 1 or 2
I used a little more time, Michael.
Yeah, if there's 1 or 2 pressing questions, we could do them now, and then we'll also have the panel discussion at the end. So any comments or questions that are burning for Lee? John?
A question concerning possible sources of failure. What about errors in the identification of cell lines? This is a problem which has been reported repeatedly in the scientific literature during the last half year or so. What progress is being made?
Yeah, so I would say that is a real problem, but that's a bigger problem in the literature and in academia. I think once, and I will tell you in the next program, Myrtle and Bev Teicher are smiling because often when the NCI takes a molecule or something into their pipeline and they test it against the cell line that supposedly is sensitive, it's completely wrong, and that happens over and over and over again. But that's sorted out early. So if that happens now, that's the end of it. We just say, you know what? The cell line's not what characterized the way it should have been, and it's not right. But once you get into pharma development, that's not really a big problem anymore. Most of these cell lines and xenografts are fully genetically characterized, and I can tell you, I'm sure pharma has complete genetic characterization of everything. So it's a bigger problem in the literature and for academia, but once it really gets into a campaign, that's not the problem.
So Lee, I preface the question and a request for a comment for you with the sort of idea that this may help us sort of set expectations of what comparative oncology could provide. But sort of thinking about the phase 2 to phase 3 failures. So to me, and I'm interested in your thoughts on that, to me that means that human cancers, where you're just trying to make a tumor smaller, are not good models for human cancers where you're trying to improve outcome. So human cancers are not good models for human cancers, is my interpretation of the phase 2 to phase 3 failure. Can you tell me that I'm wrong there?
So you're getting into an issue of endpoints, and is response rate, which is typically what we use in phase 2, predictive of improvement in outcome. And I would say we could spend 2 days just talking about that, and it's controversial, and I would say it is something that we need to be a little bit more rigorous about. Partly it has to do with durability of responses, and you could even argue that in some cases, overall survival may not be as important as the health of a patient while they're living with cancer. And so that is a very complicated issue, but I do think it does raise the point that we got to be careful about our endpoints in phase 2, and maybe that's the more important answer before we move into phase 3. And if we're convinced our endpoints are right, then we should have. And there's no question we pick bad endpoints sometimes in phase 2, and that is a reason for attrition.
So I won't ask the question now, but at the panel discussion, we should follow up on that, in that it's not just whether the tumor responds to an agent. It's also the cure that you're talking about is always in combination with other agents. So there's a big challenge for us in how to combine these new agents, and where the canine models can be useful in that, where in human trials when we do that, it takes a long time and it's challenging. So how we can use both models to inform each other in combination therapies is something we probably should discuss at during the discussion session.
Totally agree with you, Mike.
So just along that line, I mean, you can look at the data, 1 in 8 to 1 in 12 phase 2 successes is a phase 3 success. So I bring that up mostly so that we can set our expectations about what the dog can add. I mean, from my point of view, the hurdle is pretty low, but we should keep that in mind as we sort of decide how we're going to measure our success. So if I was to tell you I'm going to be right 1 in 8 times now, and we don't think of the data that Lee presented, we'd say that doesn't sound all that helpful. But if we say that right now the human-to-human scenario is 1 in 8, the dog doesn't have to do a lot more to make us feel like that's a good idea.
At much lower cost, potentially. All right, thank you, Lee. That's a great start to the morning.